Thank you, Anish. Thanks to everyone who's helped bring me here. It's very nice to come visit. I was told a few people that this building and the lecture theatre down there is where I had my very first maths lecture at university. So it's nice to now be speaking in the same building. So today I want to talk about phylogenetic approaches to measuring biodiversity. And so when we think of this idea of biodiversity, it's one way to think of it as the variety of life and types of life on Earth. And but we'd like to be a bit more specific than that, especially if we have, I'm looking to quantify this idea. Because what happens is we'll see, sometimes you can see news headlines that look like this. You say nature groups launch legal bid over wildlife loss. And in some sense, I assume that part of their bid is going to be measuring this loss and quantifying it in some way. And here's another headline I found, which I quite enjoyed, which was that the global rugby family, World Rugby, are trying to help tackle biodiversity. So everything, you thought nothing was safe from a rugby metaphor, well, there's the proof. Tackling biodiversity, great idea, but yeah. And here's another one. Accelerating extinction rate triggers domino effect of biodiversity loss. So here are these, and here's a nice tortoise here. Here are these ideas of biodiversity loss as something that we'd like to quantify. And maybe it's different or related to an extinction rate. Often though, when you look into these articles and what research is behind them, there's some things that are common in terms of what biodiversity is being measured. Often it's abundances of species. So how many actual individual organisms there are in each particular species. And more crudely, if we're just thinking about whether species are present or absent in an area, the sheer number of species, what we call species richness, is often meant to stand in for biodiversity. And it can be a reasonable idea, but give us a first view of how biodiversity is changing. But we can do a thought experiment to picture why this might not be the best measure. So my title here is Species Richness is Too Simplistic. We can think of these two islands. One island has got 9 species and they're quite varied and different. And island 2 is Fern Island, which just has 10 different types of ferns. Probably looks quite nice and lush. But we get a sense where the diversity on Fern Island is far more corralled, far more constrained than it would be on this first island. On this first island, we'd say we've got lots of different types of life. And although we have 10 ferns, that probably shouldn't give us a higher biodiversity value than island number 1, which has a variety here. And the key point here, the thing that the species richness approach is missing, is taking into account the connections between different species. And I'm talking about in an evolutionary sense. And so we can look at using an idea of phylogenetic diversity to measure diversity. So here I've got 11 species represented at the bottom of this graph. And the connections are shown between them back until we have a common ancestor of these 11 species. And if I want to take this set of 5 circled species at the bottom and give a value to their diversity, well, phylogenetic diversity as a measure says what we should do is trace back all of the paths that connect these species to the root. And then we count up the lengths, the values along all of the red edges. And in this case, we'll get a score, 22. And that's reasonably quick to do. And we can get an idea of the phylogenetic diversity score. And we can see it depends on how species are related. Because these two species, they share most of the path back to the root. Whereas this species has kind of got a bit more of a unique history going on here. So phylogenetic diversity is definitely an improvement on species richness. And one way we can use this is take an area, look at all the species we have in this particular area, maybe it's a nature reserve or something like this, and measure the phylogenetic diversity of the set. This is a quantity that we can see how it increases or decreases over time. So we may get species going extinct, and that would decrease phylogenetic diversity, or maybe we've been protecting an area and new species are coming in to our nature reserve and increasing the phylogenetic diversity of that particular nature reserve. And it depends what scale you want to look at, maybe looking on a whole country scale or continent scale as well. And I'm interested in what happens when extinctions occur. So extinctions are a big deal in conservation. Once you lose a species, you're never getting this back. And we want to be able to quantify what that loss is. We know it's a bad thing to lose a species, but is every species being lost as bad as each other? So we can put some numbers to that question. And we can think about particular extinction events. Maybe we have K species out of a total of N species that survive some extinction event. Well, maybe there's a difference of outcome depending on which K species we choose, or which K species survive the extinction event rather. And we can ask which K species would we select if we were wanting to select those that would retain the maximum PD. So you might think of this as which species do we want to give conservation attention and funding towards because they represent quite a diverse breadth of the variety of life that's in a particular area or country. And part of the reasoning for this is simply that we don't know much about a lot of species and how a lot of species work and how a lot of the interactions in the environment work and what the benefits to humans are from different species. So our best bet to keep the healthiest ecosystem, to keep the best benefits for all species and humans, is to have a wide variety of species that stay alive. So if we come back to this example, I can see that these 5 species, if I'm limited to choosing 5 out of these 11 species to focus my attention on, this is not going to be the best overall choice. This is not going to have the highest phylogenetic diversity score. Can anyone see one that you would exchange out and swap out? Anyone want to guess which one? One of these two, they're quite closely related. So if we took one of these away, we could replace it with, say, X11 here. And so we would lose this small red line with length 1, but we would gain 5 along here. And that actually would be one of the maximal possible ways to do this. Turns out there's 84 options if you're choosing 5 species here, and you have these 11 species with this relationship. One way you can think about that to maximize the phylogenetic diversity is, well, there are 2 things going on here that made me notice it's not the maximum possible. One is that we're missing out on being contained in this whole area of the tree. And the other is these 2 are too close together. They weren't adding much. The second one wasn't adding much after the first one was already there. So what you can do is we could break that tree down at a particular height, they broke it into these 4 components. So this one's just a single vertex here. And if we want the breadth of species in our collection, we need to choose at least something from each of those 4 components. And if we chop it at a lower height, where it now breaks it into 7 components, so we wanted 5, we had to go 1, the closest we could get above and below, which was 4 and 7. We don't want to choose the 2 species that are in any one of these groupings, because they'll be too close together. And so I was able to show that the sets that maximize phylogenetic diversity have exactly this property. They're spread out enough in the components, and they also don't bump into each other in these shorter components. And I mentioned there were 84 there because based on that characterization, I can come up with a nice generating function, come up with those sort of numbers. But these numbers can actually get quite big. So this is on a very balanced tree. So you can imagine you're going exactly breaking into 2 daughter species at each possible height, exactly balanced on both sides. And you can see, well, okay, if I've got 16 species, I wanted to choose 10, we have 1,800, and then it jumps up. If I have 32 species, I want to choose 21, we get 9 million, and then it gets into a very larger number already. Yeah. And so that gives, these sort of numbers are a bit daunting, because you can imagine going to someone who's going to implement a conservation strategy, and they say, great, we've sent away you, the mathematician, to tell us, find us a set that is as diverse as possible from these 32 species. We can put $21 million out and 1 million to each of these species. And I would say, oh yeah, sure. You just can choose any of these nearly 9 million possibilities. And they go, oh, thanks. That was really useful. So we need some way to kind of distinguish between these millions of possibilities to get down to a manageable solution. And the way to do that is by incorporating further information. So here I've got the same tree, except now down here in this row, I've added a number that represents the endangerment level of these species. So these species labeled 1, they might be ranked least concern by the IUCN. Whereas number 6 here, that might be extinct in the wild, and the 5s might be critically endangered, say. So you could think of that the higher the number here, the more endangered the species are. And by using this idea of breaking the tree down into those groupings, is an algorithm that I developed where we can find, in this case, the unique set, which is species 1, 3, 4, 6, and 10, which is a group of 5 species that has the maximum diversity, but contains the most endangered species amongst those sets that have the maximum diversity. So if we were going just on endangerment alone, we probably would pick X9 instead of X4. But then we wouldn't be spread wide enough. We would be missing out on all of this phylogenetic diversity if X4 went extinct through lack of being looked after. So this incorporating further information is a key way to help us distinguish between those possible large numbers of maximum PD sets. But I'm going to spend the rest of the talk talking about another approach, and another approach to finding these secondary values. And we have this concept called a phylogenetic diversity index. And so this is thinking about a phylogenetic tree and trying to measure evolutionary isolation. In particular, we're now looking at a species by species focus instead of a set of species. So instead of saying, what's the set of so many species that maximize the diversity, we want to say which individual species are more uniquely placed on a phylogenetic tree. And this kind of comes into being important because if we think about how conservation is done, there are plenty of things that are done on a single species basis. So we might be looking after a particular environment that one species needs to survive, or things like these breeding programs. So here's a kakapo, and there's been quite an inventive use here where these fake eggs have been developed that can even have movements, small movements happen inside these eggs. And so when there's a nest with an egg in there, they sneak in, they swap out the egg for one of these artificial eggs and incubate the egg in a controlled environment so that rats don't get to it, but still maintaining that kind of parental behavior that we want the bird to remember how to do. And so this egg tricks the bird into thinking about, okay, I'm still in a good place. I've still got this egg. It's still moving now and then. And then once it's close to hatching, they swap it back out. But the point is, this is not easy to develop or cheap to develop or quick to develop. So these resources that need to be used in conservation practices sometimes need to be quite concentrated. And so understanding where they'll be best used does have some importance. So here's kind of an example from the literature where these researchers found a way to rank amphibians by evolutionary distinctiveness. And so we end up getting a list saying that these I think the top 10 or so, these are the top 10 most distinct amphibians. And this list continues down and they have values there. And so if we were looking to just prioritize based on the evolutionary distinctiveness, we would start to look to these species for where to go. And I've got a picture here of the olm and the purple frog. So they can be quite distinctive. The Mexican burrowing toad I have not found a good picture of. I think it lives, either it's extinct or it lives so long in the mud that there's no really nice picture of them. But so what I'm going to talk about from now on is talking about what methods are used to rank these species. How do we come up with such a ranking? And are these good methods, or do they have kind of issues that we need to think about? So the first method is called the Fair Proportion Index, or FP. And this involves giving a number to each species based on its unique history and some fair proportion of the shared history it has going back to the root of our tree that we have here. And so if we focus in on X6, we'll see that this branch here represents the unique history belonging to X6. And so this value of 3 goes into our calculation. This branch with length 2, well, that's split into 2. There's 2 species that embody the features that were developed, or the evolutionary change that was developed along that branch. And so we'll split that value, divide that value by 2 and add it on to our running total. And then this edge 6 is then shared between those 3. So the fair proportion that we get would be 6 divided by 3. And so we add these up, 3 plus 1 plus 2, and we get a value, 6 units. Usually this is, if we have a time tree, it would be evolutionary, say 6 million years of evolutionary history. This one is not, so yeah. And we can do this for all the species. Work out, and now we have a ranking that says, well, the fair proportion method tells us X7 is the most unique out of these species, and X2 is the least unique. But that's not the only way we can do things. The main alternative is, and really the only one that's implemented in the literature apart from fair proportion, which is by far the most popular, is the equal splits index. And the idea here is that we're not going to care how many species are on this side of the tree or this side of the tree. Whatever evolutionary developments happened along an edge, they're going to be allocated half to one branch and half to another. And so if we think about evolutionary history for X1, the equal splits would say, this is unique. This edge again gets split by 2. And now this edge gets split by 2 and then 2 again. So by the time it gets to X1, we're left with a quarter of the length of 3. And here, this branch with length 4 gets split once into half, again into quarters, and then here into eighths. And so those are the 4 values that go into measuring the equal splits score for X1, species X1. And again, we repeat the same process and we get values, but they're different values. So there's a question immediately of, well, do my conclusions depend on which index I choose, or is it just kind of a slight adjustment? And one thing we can notice is, if I look at the X4 and X6, fair proportion prefers X6, whereas equal splits prefers X4. So it's these 2 species here. And so keep that in mind, that these, it's not just a stretching or skewing of the numbers. They can turn up in different orders as well. So one thing that's nice here is instead of having a table of numbers, I like to think of these scores in terms of vectors. And that's nice because we can use all sorts of mathematical ideas that come from vector geometry. And we can think about the relationships between them on a kind of a spatial level. And because they're vectors, we can do things like adding them or adding proportions of them together to get new diversity indices. So perhaps it's the case that we have fair proportion and we have equal splits, and there's other ways of doing this, but they're just kind of some sort of combination in the middle between these two. But we'll see that's not the case usually. Here's a diagram. I've got a very small tree. And when we think of those vectors, we can think of the endpoints here. So if I was, there's 2-dimensional space here, and this point here represents the fair proportion index. That point is our method. And the equal splits method is here, this one. And every point within this boundary just represents another potential method that we could use to describe how the edge lengths from this tree should get allocated to those species to kind of talk about their evolutionary history. And these two are reasonably centrally located. As we get closer to the edge, we are making stranger and stranger choices in a way, less biologically reasonable ones. And when we get to the boundary here, what's happening is we're saying that there's going to be a 100% split on one side of a vertex and 0 to another. So that's saying everything along here would be saying that this edge, even though it was ancestral to X4, we're going to allocate all of the evolutionary change, all of the developments there only to these 3 species. we're going to ignore the fact that X4 is a descendant. We can't totally take this edge away from the species though, so it does get some value. So this is a boundary condition where we run into no longer being sensibly on a biological level. So these 2 points, they're distinct, but how big is this difference? And it turns out that here's a nice contrived example where they're completely ranking the species in opposite orders from one another. So, okay, which one do we choose then? One equal splits is saying that we must prioritize X5 over X4, I think over X6, 3, 2, 1, and fair proportion is saying no, X1 and X2 is best or the most unique, and then X3 and then down to X5 is actually the least important here. So understanding the properties of these different approaches is what I'm interested in, so that we can answer questions like this: which ordering should we be taking? And there's 4 key points here, so that we can be sure that our method is stable and accurate and actually kind of gives us the information that we want it to. And I'll go through those with you. So we have, well, I guess it's best we'll just go on. Largely, we're concerned about changes in the tree. How do we understand changes in the tree? But this first one, and this has been worked on recently by my supervisor Charles and Magnus Bordovich, that we have to consider the relative positions of different species in the tree. So in this tree, it's very balanced. So every species will end up having the same fair proportion score, so regardless of which 4 we choose. But these 4 are all clumped together. If we were just choosing these 4, we would miss out on everything on this whole side of the evolutionary branch. There's a much greater distribution here on this side if we choose the species in a more spread out way. And so sometimes this can happen where the diversity index is pushing you towards choosing some species in one particular corner of the phylogeny, whereas we still need to keep in mind those other ideas from earlier that we want to spread out those species that we consider. The other thing to keep in mind is that trees are in a constant state of change as people learn more about the relationships between species, as people learn of more species, as extinctions happen, as former species are reassigned into different new species and subspecies kind of being promoted to species level. And so one thing to ask is, are we going to disrupt our processes if we move from a tree like this, where we have the polytomy, into a more resolved tree? And got a result that says that the fair proportion index is the only one that is not at risk of large changes or abrupt changes as we introduce small edges. It's framed the other way in the sense of going backwards from here, contracting that edge to this tree does not cause a change, but in practice it works both ways. And so that's a bonus for fair proportion. You think, well, okay, if it's the only one that has that, that might be enough to say fair proportion is better than equal splits. But then along came this nice result by these 3 authors that said we can find scenarios where when species go extinct, when we recalculate the index on the new tree that's now missing a particular species, say X1, the ranking order that we get, even though we're using the very same method, the ranking order can be completely flipped upside down. And so that doesn't suggest that we have a very robust thing. So these authors showed this effect for fair proportion. So we have this kind of question now. Is fair proportion the only one that suffers this? So here in this example, I've got when X1 is still alive, is still part of the tree, we think they become more unique as we head from left to right. If X1 goes extinct, then they become more unique from right to left. And the result is that any topology you have, any tree structure you have, we can find lengths and extinction events where this reversal is possible. And so one result that I had in a recently published paper is to say that it's not just fair proportion that suffers from this problem. So every diversity index, if we go back to thinking about that, that space, that kind of trapezium-shaped space that I had, those spaces of indices, every possible choice of method that's not corresponding to one of those ones on the edge has the same problem. And I argue that those indices on the edge are so biologically unreasonable that we can say that every reasonable diversity index method is just as lacking in robustness in this sense as fair proportion. So this is not a reason to kick fair proportion off to the side. And in a kind of a related fashion, I want to talk about what's called species inflation by some people. So here's a picture of a giraffe. And giraffes used to be one species, and now I think they're 4 or possibly more, when subspecies were promoted to species level. And kind of intuitively think that if this species was endangered to a certain extent, by breaking it up into more species, we would could only increase the endangerment level of the new giraffes. But fair proportion, or all of these measures really, would give those 4 giraffes smaller scores compared to the rest of the species in the tree than what the single giraffe had on its own. Because now a lot of those paths back to the root are going to have to be shared between 4 more species compared to when they were all just assigned to the giraffe itself. And in some sense, this is actually the more common change. We hear a lot about extinctions in this field, but the more common change is the species being added. There's 2 reasons for that. It's quite hard to confirm whether species are extinct or not. And people are reticent to do it. There's well-known examples like the takahe, which was declared extinct and then rediscovered again. And so if species are declared extinct, that often means, well, no conservation funding is going to go to an extinct species, but some might go to preserving the types of environment that that species would need to live in without it. And there's also kind of an effect where the more a particular species is studied, the more often it looks like it might be some different species. So this is an asymmetric effect across the tree of life. The more charismatic and well-known species often get split more so than those that are less so. And so my way to kind of thinking about adapting this diversity index approach to take into account is to have another look at how fair proportion and equal splits work. If I'm trying to think about how this edge E is shared between these 7 species, fair proportion says I'm going to look below this vertex and understand exactly that there are 2 on this side and 5 on this side, and share out the value of this edge accordingly. Whereas equal split says, I don't care, I'm going to give half to share between these 2 and half to share between those 5. And so fair proportion in some sense is very long-sighted. It looks at the entire tree below this point to make its decision, to make its allocation choices here. Whereas equal splits is extremely short-sighted. It takes into account nothing of the information past to be. And so perhaps we can have some method that's in between these two. You take a particular window into the future that allows us to see what sort of speciation events were happening on either side of the tree below this particular edge, and then share out information or the value of E accordingly. The other approach that I've considered is, especially when it comes to the species inflation kind of idea, is that when new species are added, they often have very close living relatives. And so the additional edge will be a very short one like this, or even shorter perhaps. And so there's a sense in which we could chop the tree and calculate the fair proportion values for everything above that tree. And you would get every species that's connected below where you chopped would just get the same value. They're very closely related, so they'll have similar values anyway. We might as well have a more robust approach, just give them the same value, and then we can use other information like conservation status, or maybe the benefit to cost ratio of conservation action in that area to choose between those two if necessary. And that's it. That's all I was going to say. So thank you for having me again.